So in case of phytoplasma, mainly it is using various mechanisms to cause different symptoms. Mostly it is symptom mimic like viruses. So as you said, there is no any control measure for this, at least for Sesamum pylori, this phytoplasma disease. You cannot use resistant source, or even you cannot apply chemicals to control the disease. So somehow we want to control the disease. There comes the approach. So first thing is we need to characterize the group. At least in Tamil Nadu, throughout the world, as I said earlier, there have been 34 groups. So we wanted to know what are the groups present in Tamil Nadu. So that's why we have chosen the topic, and we allotted one thesis topic to assess the geographic distribution of this phytoplasma disease in various crops in Tamil Nadu. Then we wanted to characterize all the groups, and we wanted to develop on early detection kit. You may say that symptom is visible, why do you want to develop the kit? But at least if you develop the kit for early detection, it may be good for recommending some management measures to contain the level of incidence. In some other cases, serials, the symptom may not be visible, it's asymptomatic. So in that case, it can be useful for at least scientific point of view to diagnose the disease. So characterization, diagnosis, and finally comes the management. We tried different methods, chemical, physical and biological, and even resistant gene, the gene bank from the NPPGR. We got lot of resources, around 6,000 accession we screened for, at least for Sesamum. None of them are working again, giving resistance to the disease. So the approach is to develop at least one resistant variety. So here you could see in Tamil Nadu, so it's occurring almost 16 different. So far whatever we have documented in major crops like chili, vegetables, apart from the Sesamum, other oil seeds like groundnut and vegetables, aromatic crops and coconut also. There are many, including various weeds also getting affected with the phytoplasma. So that we have documented. Next we have characterized the organisms. So we found that out of 34 groups, 4 groups are present in Tamil Nadu. Yeah, only in Tamil Nadu, not in entire India. In Tamil Nadu we found 4, there is 16 as are 1, 2, 5 and 6 group with 9 subgroups. So that is one thing. And you know the symptom, it can cause different type of symptoms. So the challenges in working with the phytoplasma, it is non-culturable one. We cannot, that is the difficult. We made some attempt, but we couldn't able to culture this organism. It is unculturable one. And because of this obligated nature, it will colonize the flowing region. Still it is very complex to isolate and characterize the organ. That is one thing. Another thing is it is maintaining tri-traffic interaction. Like a virus, you have to study about the host, insect vector and the pathogen. When it comes to vector, again it is a complex. Previously people used to say there is only one, mostly like the hemipter pest, leaf hopper or plant hoppers are the vector of this phytoplasma. Previously people used to say there is only one vector responsible for the particular phytoplasma disease, but that is not the case nowadays. If you go to the field, you could find a complex of phytoplasma. So it is a mixture of phytoplasma, not a single phytoplasma causing this same disease. Various phytoplasma spreading the same disease in the field. Say for example, whether it's a brinjal or sesamum or whatever it is. So that is another thing. And there is not much molecular work to dive into this phytoplasma to study its characters or the interaction between the host to pathogen. So these are all the various challenges working with the phytoplasma. Still it is keep on continuing. There are stalwarts working on the phytoplasma, but still it is very challenging too. So if we want to understand the pathogen and to control, we should link study the genomic data and the practical challenges in detecting this pathogen well in advance, the field condition. So for that, we decided to develop one RP assay. Mainly we thought of developing some LF based kit, but if we end up with RP assay for there are advanced PCR methods are there, but they are time consuming. But with this method we can diagnose the disease within 30 minutes. You don't have to wait for 3 or 4 hours like PCR. That too with a crude extract, you don't have to extract DNA also. So here you could see the sensitivity of this method when compared to PCR. Even if you extract DNA, you can also use the DNA for this RP assay like PCR. The results will be same. But when you extract the crude and directly if you use for PCR, it won't work. But for RP assay, you can get the result within 30 minutes. It's just a thermal amplification method. You don't need sophisticated instrument or laborious or time. There is no such a case. Okay, within 30 minutes you can detect this disease by using this method. So this is one approach. So next one is the management aspect. okay, something is missing here. So probably you might be knowing about the gene for gene hypothesis. So in the biological system, everything is governed by gene, right? So here we wanted to study, if you want to study the interaction between the pathogen and host, there must be some resistant gene or susceptible gene in the plant system, and correspondingly there will be some virulent factor or virulent gene or pathogen factor in the pathogen. So we want to crack down that one. But here in case of Phytoplasma, the CEFIPRA-FUNDED effector is the main culprit causing the disease in various crop plants. There is one thing, correspondingly if you see the plant system, I will come to the later, because if you see the symptom of this Phytoplasma, it will look very unique. It will convert the floral system into leaf-like, it will modify the floral parts into leaf-like system, or you may find some malformations. It's all because of this. It will go and directly interact with the mat spark system, which is highly essential for the development of the reproductive structure in the plant. That's why all the time when the plants are getting infected with the Phytoplasma, it will become sterile. They will directly go and infect the mat spark system in the plant system, so they cannot be able to produce flowers or fruits or whatever the grain. So but these genes are very essential. They are all the susceptible gene. But if you go and touch or edit the mat spark system, the entire the plant system will get collapsed. Again, people started working on it. Finally, they found that there is a shuttle protein between the mat spark gene and the effector protein in the pathogen. There is a protein called RAT23, which is acting as a shuttle protein between this pathogen protein and the plant protein in the plant system. So they have decided to silence that particular gene, so that the pathogen protein cannot recognized by the plant system for a compatible interaction. That's why I was talking about gene for gene hypothesis. When there is a compatible system only, the plant will produce disease or symptoms. If it is incompatible, it will become resistant. So the thing is here, the RAT23 protein in the plant is the susceptible factor responsible for causing the disease, Phytoplasma disease in the plant system, whereas the effector is a virulent factor in the pathogen. So we cannot do anything with the Phytoplasma effector protein, but at least we can restrict we can knock down the susceptible factor in the plant system. So with that approach, we have identified and characterized the CEFIPRA-FUNDED effector protein in the Phytoplasma in all the groups, and simultaneously here we took the sesamum as a model plant here. So we have quantified the effector expression in the infected plant. The level of expression varying after infection from 15 days till maturity in all the plant parts like stem, leaves and flowers also. It is having the expression, the Phytoplasma expression is there. So as I said earlier, there is no any method to control this particular disease. So with the only approach, genome editing only, we could be able to develop the resistance in the plant system. So we have identified the target gene as or susceptible factor as a RAT23. So here we have developed the guide RNA for this particular protein, and with the help of genome editing, we could be able to transfer the guide RNA into the plant system. So that will give resistance. When there is no susceptible factor or particular corresponding gene in the plant system, the pathogen cannot be able to recognize and it will cause the disease. So that is the idea. So with that idea, the work is halfway still. We could be able to develop the tissue culture plant, and we identified the target gene, and we have transformed the gene. So it is still under evaluation. We need to go for the phenotypic assay and through transmission insect assay, as well as we also developed the clone for that. CEFIPRA-FUNDED effector clone is there. With that also, through agro-infiltration method, we can prove the pathogenicity, either through agro-infiltration method or through the conventional insect transmission assay. We are trying to evaluate its efficacy or its resistance under field conditions. So that work still is going on. So this is the only way to work with Phytoplasma. First thing is we need to characterize it, and we should develop some early detection kit, and we should study and characterize the effector protein present in the pathogen system, because that is the main culprit or the factor responsible for causing the disease in the plant system. And the only approach I could feel the genome editing. Genome editing would be the option for developing the resistance. Thank you anyway. That is the thing I want to convey here.
Long live India and France friendship.